So thanks a lot to the organizers for giving me this opportunity. This is not a lecture I usually give, so it has been the occasion for me to think about what I have been doing in the last 10 years or so, or even more. And I'll share with you something that is a bit of formalism and also a lot of speculation or more philosophical thinking about the evolution of collective behavior. So this is, I think, some step towards combining fields that have been developing a lot in the last 20 years, so from, or more or less. So on the one side, on the side of physics, you have the physics of collective behavior have witnessed great advances. And these studies, which I was trained in, are often very quantitative and often, when applied to biological systems, they try to look at the mechanism that is related to collective behavior. On the other hand, from the point of view of evolutionary biology, there has been also a great effort in trying to formalize what collective behavior is, how it can evolve. And this has led to the development of another set of models and approaches that actually ask rather how this collective behavior changes in time through the process of natural selection. So this has sparkled these approaches in evolutionary biology, some hot debates about exactly to what extent a certain theoretical framework can be shifted from one set of organisms to another. And I'll try to touch upon this existence of different time scales, different description scales of biological systems that actually makes the description of evolutionary dynamics a bit tricky. And still, I think this is pretty open in terms of understanding how collective behavior can evolve. So let me start just with an image. So what do I mean by collective behavior is something like what you have seen in the past days. Basically, here you have a population of limit cycle oscillators that are globally coupled. And at some intermediate coupling strength, you have that even though each oscillator in isolation has only a limit cycle, here the mean field, that's the red cross, the average of all the oscillators that you see projected in a plane, has actually some strange behavior, well, chaotic, or it can have some, let's say, low-dimensional complex behavior or high-dimensional complex behavior. But what is interesting is that basically, depending on the interaction between different elements, your macroscopic description of the system can sort of decouple from the microscopic description that you would have if you just took one oscillator and took it out of the population. So this is more or less the picture that a physicist, I guess, has of collective behavior is just one example. And now let me go to some examples of collective behavior that are encountered in biological systems. So you have seen last week some examples of physiological oscillations. Here you have the inner ear bundles. You have heard Dolores speak about this system, and Pascal, one of the organizers, is also a specialist in this kind of oscillations. And so these are oscillations that emerge as properties of some dynamical features of the single cilia. And you have similar oscillations a bit all over in physiology. I mean, they have been discovered in many tissues. For instance, pancreatic beta cells undergo similar kind of oscillations. And of course, neurons, even though in the case of neurons, actually is not good that you see oscillations at the population level because they are typically associated to epileptic seizures. So this is, let's say, within one organism. But this is not the only domain in which you see these collective oscillations that derive from the interaction of many oscillators. You have here two examples. There are more, but just to give you an idea of metabolic oscillations in yeast cell populations. So this kind of oscillations that you see here, this is fluorescence, this is, I think, oxygen content in some stirred reactor, continuous flow reactor. And these oscillations are evident at the population level. These are clearly some collective behavior. What is exactly the function of this collective behavior? It's sometimes known, sometimes not. For these oscillations that are relatively slow, they are of the order of a few tenths of minutes, what people have shown is that they are coupled to the transcription profile in the population, which means that they offer the possibility basically to decouple the transcription of genes that are in the mitochondria from those that are in the nucleus. So this is an hypothesis of a putative function for this kind of oscillations. Whereas for these oscillations that are too fast to affect gene transcription, these are like 40 seconds, they have been described extremely well and in extreme detail from a biochemical viewpoint, but nobody really knows whether they have a function or not in biological terms. So why are they there? Are they used for some purpose? Now some other examples. This is what Hugues Chatet has been talking about. So for instance, flocks and swarms, you don't see them only in the world of macroorganisms, but even in microorganisms, you see this kind of structures. This is an example from myxobacteria that are swarming, and reminds a bit what Vincent and Pascal, I was not here at your talk, but I guess that you must have shown something like this. So basically you have individual cells that are rod-shaped that form these swarms. And actually these swarms are important for them to acquire their nutrients because they basically chase through the formation of these swarms. So Starlings is not really clear what's the function of this aggregates. It might or it might not have really a biological function from an evolutionary viewpoint. And these ones seem to have a function which is not easy to quantify. Now some other examples are related. So the previous examples were all phenomena, collective phenomena that emerge basically on time scales that are shorter or of the same order of magnitude of the reproduction of the individuals. However, you can have some collective phenomena also in growing populations. So for instance, this is a beautiful picture that was taken by Paul Rainey, who is sitting out there, of wrinkly spreader variants of Pseudomonas fluorescens. So these cells have a collective function in the sense that they colonize the air-liquid interface and they form a mat that actually helps somehow the other cells of the same type to float and therefore to have access to oxygen in experimental microcosms. And another example are biofilms. So this is a special case of a biofilm. There are biofilms that are composed by more than one cellular type. And also this seemed to confer an increased resistance of cells to stress, and therefore this could be the reason why they have been favored by natural selection. And I'll get back to this towards the end of the talk. Similarly, you can have that even not in the presence of some substrate, you can have that cells clump. For instance, these are yeast cells that are flocculating, so they clump even in a mixed, well-mixed environment. Or they can form flakes, these are called snowflakes, because the cells do not divide. If a given selective pressure is applied, actually this kind of configurations can be favored. And actually this one has been obtained through an experiment of directed evolution in which people were selecting for things that were sedimenting fast. So basically they knew what was the selective pressure that they applied to the population, and what it came out is that some yeast cells learned, or they discovered somehow to divert some of their pathways so that they form a multicellular aggregate. Finally, my last example are things that seem to have clearly a motivation, a function at the collective level. And these are multicellular differentiated fruiting bodies that are observed in aggregating microbes such as myxobacteria again and slime molds. So in these two microbiological systems, populations of cells live most of their life at the unicellular stage, and then they aggregate to form these multicellular bodies that actually produce on the tip spores, and it is thought that the function of these little mushrooms is to let spores disperse further away, so basically out of the region that is this one that has been depleted in nutrients. So this is something that everybody would probably agree with. Still, one has to explain how do properties that are a feature of a collective of cells evolve in a world where usually selection acts at the level of the cells. So why are the structure not disrupted quickly by the appearance of cells who do not contribute to the endeavor of building up a multicellular body? Yeah.
Excuse me, what would be the characteristics of this?
So the height, they are a few millimeters, let's say scale 1 millimeter high. And the number of cells that are within the fruiting body depends on some parameters. It's, let's say, the order of 10 to the 4th, but it depends on the density, humidity, plenty of things, and signaling between the cells. So there's no really quantification of how the size varies with these parameters, but more or less is in this order of magnitude. And I forgot, the question was, how big are these fruiting bodies? So now I'll go to the blackboard and just introduce you to some concepts that come from evolutionary biology. I will start very simple. So is there any evolutionary biologist among you? One. Okay, then you will correct me if I say something inappropriate. But so I start out very, very simple, just repeating some of the what makes biological systems different in some respect with respect to some other systems. And basically, the main property is that biological units, whatever they are, cells, multicellular bodies, or even molecules within a cell, they have the capacity of replicating and of multiplying. So let's say that you have one unit. Let me speak by simplicity of this as if it was a cell. So this is a cell, a white cell, and after some time this gives rise to 2 cells, and after some time you have 3 cells. So this has multiplied, and the new cells that arise, they are just, they have inherited somehow the properties of this cell. And this is also a feature of most biological systems, that you have some heredity that propagates the feature of the initial unit throughout time. Now consider you have another cell with different features, and this cell at this time has given rise to 2 cells, and at this time has given rise, I don't know, to 5 cells. So if these cells are more or less coexisting in the same environment, usually people think that this cell has a higher fitness, is more adapted to the environment. In the population, looking at the population level, the population will be soon composed only of purple cells, whereas the white cells will disappear unless there are some effects of frequency dependence or well, unless it is the real world. But so let's take just this very simple schema. What makes these particles different from what one observes, for instance, in coupled oscillators, is that they multiply, so they change in frequency and in number in the course of time. In 1970, Richard Lewontin has proposed 3 necessary conditions for natural selection to act on such a system. So a system of entities that multiply and they can transmit their properties. So the first condition is that there is a phenotypic variance. That means that you don't have cells, in this example, that you don't have cells all of the same type. The second one is that there is phenotypic variation, sorry. This variance is associated to difference in fitness. So this type is more fit than this one. So differential fitness and the third condition is that fitness is inherited. So fitness inheritance. So given these 3 conditions, any system, unless some very particular cases, will evolve by natural selection. And so the fittest type, usually in simple settings, will take over other types. Now, this is a very classical view of evolution by natural selection. However, it relies on this fitness that is a concept that is a bit problematic, and many philosophers of biology still struggle with this concept because it is actually quite hard to measure exactly what one means by fitness, how much a given type is fit or adapted to a given environment. And it's also a concept that can change depending on the time scale one looks at the system. So it's a concept that is extremely fundamental in all evolutionary biology, but that is, if one wants to go to the point of comparing with some observation, it is a bit problematic. I'll later, if I have time, I'll go back to discussing whether one can actually define the same necessary conditions, but using other terms that do not involve the measure of fitness. So a second point, property of biological systems is that they are organized at different scales, spatially and temporally. So this process of multiplication with heredity can be seen on many different levels. So for instance, you can see it at the level of molecules, just replicating molecules. I don't know, RNA or anything within a cell that replicates. Second, you can see it at the level of the chromosomes. The chromosomes themselves replicate within cells. Another level of organization is that of the cell itself. And notably, cells can be of different complexity, depending whether they are prokaryotes or eukaryotes. Then above the cells, you have the level of organization of a whole multicellular body. So, multicellular bodies. And then many people recognize that there is an organization also at the level of societies. So what is typical of this different organization levels of the biological world is that every level contains the lower level. So the idea is that evolution, so historically, has led from molecules down or up to societies by integrating different units that each of which is able to replicate. Yes?
The fitness of the chromosomes.
Yeah. I have problems with the fitness, but let's say that one can think that in some primitive world, chromosomes were replicating independently of the cells. Yeah, in this case, I'm not exactly sure what would be the fitness of chromosomes. But for instance, it's evident with cell and multicellular bodies that you can compute a fitness just in terms of reproduction rate of cells, and you can compute a fitness in terms of reproduction rate of the multicellular body, for instance. And actually, in a very influential book, Miner-Smith and Eustasmary, so let's see if I can spell it correctly. So in a book of 1995, they have identified 7 major evolutionary transitions that had allowed to go from one level of organization to another level. And the new level was containing the old level. And the old level, after the transition has occurred, is only able to replicate if it is in the context of the higher level. So this is what is called major evolutionary transitions. So Major evolutionary transitions, there's a number of this. For instance, the origin of the eukaryotic cell by endosymbiosis. There have been probably some really keystone events in the evolution of biological complexity. However, each one of these transitions inherently carries some problems. If you think that you are going from a situation in which you have single entities that reproduce, a bit as I said before, and then these entities at a certain moment they become part of a larger entity, and this entity itself reproduces. Now, the problem of this larger entity, you can think of a multicellular body, is that let's say that within the multicellular body, you have a mutant cell that reproduces faster than the white one, and then this mutant cell will eventually invade the whole multicellular body. If this mutation is somehow creating problems to the coherence of the multicellular body, then the multicellular body will simply disappear and die out. And this is actually what is known as, has many names, is the same in evolutionary biology as it is in economy, like the tragedy of the commons is known, or free rider problem. In general, it is related to the conflicts that arise between the existence of a higher level of organization that contains units that are able to self-perpetuate and to invade this multicellular body. If you think of cancer, this is exactly what happens. You have that certain cells are able to grow unchecked, and eventually this is certainly not good for the multicellular body. So theory predicts that basically this tragedy of the commons should be extremely common. So the tragedy of the commons is a term that comes in reality from economics. And so it was Hardin in 1968 who pointed out that basically every grouping or every organism every grouping that has the possibility for this free riding is basically doomed to fail and to just disappear face of either natural selection or, in the case of economics, just the spread of cheater types. So this kind of point of view has been somehow integrated also in evolutionary biology. There are actually several ways this has been tackled. What I'll introduce here briefly is the formalism of evolutionary game theory that basically describes this situation by saying, so I have a group, a big group of size n, and within these groups I have two types. So the, let's say, good ones are called the cooperators because they produce something that allows the multicellular, the group to survive and to proliferate. Whereas these ones that can invade this group are called the defectors or cheaters, but I like cheaters less, especially because it starts with the same letter as cooperators and as an index is not very good. So very simple models that basically provide what will be the probability of reproduction of this type versus this type are called public good games. So I will just introduce you to the simplest of these public good games that is a linear public good game. This game goes as follows. So let's say that I take 10 people out of you, and then each of you has €1. And you can either invest or not this euro in a common pool. So you take your decision, I take the euros that have been invested, I multiply them by 2, and then I divide what is in the common pool among all the people, irrespective of what it was their investment. In the context of economy, basically this will give a payoff. A payoff corresponds roughly to a fitness in a biological context. And if you are a cooperator, your payoff will be the benefit that you get from receiving a share of the common pool. And this is simply proportional to the fraction of cooperators that are in a group of size n. So B, it was 2 in my case, m over n. This is the fraction of cooperator in a group of size n. And you have to take away the cost of the money you have put in the common pool. Whereas for the factors, you will have that they just get the benefit and not the cost, and they don't pay the cost. Obviously, what is better to do? To be a defector. And the fact is that everybody defects, nobody gets nothing. So this is the same kind of paradoxical behavior as it is simplified also in the public good game, in the prisoner's dilemma game, of which you might have heard. That is just the 2-player version of this game. So of course, this is always larger than this as soon as there is a cost into the collective action. And therefore, the prediction is that the factors should always invade, and people should not ever observe any cooperative behavior. So just to be consistent with what I'm doing later, this payoff of cooperators can be actually divided into 2 parts. There's one part that is due to the action of the player itself. And this is B over n minus C. So this is the benefit that comes to you by the fact that you have invested €1. It's a direct benefit that you get from your investment. Plus, you have a part that is, so to say, a social part. So this depends, sorry, there's a B here. This depends on how many other cooperators you meet within the group. And what people have pointed out is that when you go from the case in which you have one single group of size n to a population of groups of size n, what matters really is just this, basically the probability that a cooperator or a defector meets a certain number of cooperators or defectors in the population. So in this case, you have a population of groups, and each of the groups can be composed by cooperators and defectors. And therefore, cooperation will only be stable in the population if P of C is larger than P of D. And now if you call EC the probability that a cooperator meets one cooperator in that pardon? Yeah, because this is the average. This is sorry, it's a function of m. Well, m and then let me write it down properly because I'll need it later. Yeah, it's not the, sorry. Let's call it small p d of m and n. So this is what happens within a group of a given composition. this one. And this PC, big PC, is the function, is the average over the whole population of groups. And basically, this is the difference between giving a description at the microscopic level within the group and giving an evolutionary description of what happens at the population level. So you average over all the groups. And then you will have a certain probability that a cooperator meets a number of meets one cooperator in the group of size n he's in, and there is a similar probability of a defector meeting a cooperator in the same group. And then if you average over the population, what you get is a condition by which C can be larger than D. And this condition depends on these probabilities, basically how probable it is that you meet somebody who is like you in a given group with respect of meeting somebody who is different from you. Of course, in order for cooperation to invade, you need some underlying mechanism of composing the group such that basically white guys will always meet or will meet with higher probability white guys than purple guys. So basically what you get is a condition that is written in this form plus EC more ED over N larger than C. And this is also a classical formulation in evolutionary biology in which you have a condition that tells you, given a benefit and given a cost of your cooperative action, what is the coefficient that you need in order for the benefit to be sufficient to overcome the cost on average over the whole population of groups of a given size n. So this is some statistics you can compute on the population once you know how the groups are composed. It's just you just count how many cooperators are on average in the groups. And so in some one special case that has become really a classic in evolutionary biology is the case in which this quantity measures how much cells or if you want entities of the same kind are related. So this is called the relatedness, and the so-called Hamilton rules which is not particularly surprising for physicists, but it has been very fundamental in sociobiology, is that when the benefit multiplied by the relatedness is larger than C, then cooperation is stable. And actually, this relatedness, this has been used for social insects to explain why in social insects that have a haplodiploid life cycle, therefore ants, worker ants, are more related to their sister than they are to their mother, then it is easier to have cooperative behavior simply because when you help your sister, you are helping the genes, your genes in the other organism. So this can be, some people call this R as well and generalize relatedness to different cases. But what I wanted to say here is that actually you can get this, what is really important and has been really stressed by Flechter and Döbeli, who have derived this formula, is that what matters is the assortment that you have within the population. Döbeli in, forgot. 2009. Yeah. So what is important is to know, if you have groups of fixed size, what is the repartition of your particles within the group? Of course, depending on the model, you can partition particles in different ways. And for instance, if you have that your groups, macroscopically defined groups, emerge from a process of diffusion in the space, then you will have that basically spatial structure will provide or can provide sufficient assortment for cooperation to be favored. And actually, all the solutions that have been given in this context for the evolution of cooperative behavior are more or less concerned with affecting this, basically this quantity. And they have often been derived in the case in which groups already exist. So people already know what is like the macroscopic level of description of the system. They know the size of the group, and then one can compute the statistics. Yes?
So for this topic game, so at the level of the player, if the game is really blind, so there is no solving of this problem, so you have to be effective, right? And so
well, within each group, yes. But at the population level, you can also have that cooperation increases.
So I mean, for an individual player, you need to have some information about the distribution of the whole group around.
Now, imagine so the question is, how do you reconcile the fact that within each group, basically, the solution is always to defect, with the fact that at the population level, you can have that cooperation wins. This is sometimes known as the Simpsons Paradox. It just comes from the fact that the way you group things matter. So just an extreme case. Imagine that white units are able to, are grouped, I mean, maybe actively because they recognize, but just maybe passively. They only meet white guys. And the purple guys, they only meet the purple guys. Then at the population level, you will have that white wins.
But so in the case of active recognition, you need a transfer of information?
Yes, yes. This is one of, this is typically called green beard. It's, yeah, there are a series of names. Anyway, a green beard is exactly this idea, basically, that some player, a cooperative player, has advantage in carrying a green beard so that other cooperators can recognize him as a cooperator and only play with him. So actually, if you go down to like microbial populations, some cases of green beard are known, but you don't really need cooperation, really transfer of information in the sense of molecules that have evolved with the aim of transmitting this information. But I'll go back to this later. So basically, what I wanted to stress is that assortment is really the key point in determining whether, when you average over the whole population, cooperation can invade defection. So what one wants to look at is, in general, is this quantity. So what's basically the payoff difference between cooperators and defectors when averaged over the whole population. And then if one wants to go ahead in time, so what I have described you so far is basically a snapshot you can take at a single time. Once you have your population already divided into groups and that you can look at and you can compute the statistics on. But then what happens to this in evolutionary times? And this is described in this context, in the context of evolutionary game theory, by a very simple equation that is, well, extremely simple in this case, in which you only have two strategies, two possibilities. You are either a cooperator or a defector. Let's say that cooperators have a frequency x in the population. And then what rules the evolutionary dynamics is the so-called replicator equation. That writes like this. So the variation of x in time is x times the probability, so probability of being cooperator times the probability of meeting somebody who is not cooperator times the payoff difference of cooperator minus the factor. So if you want to read an introductive book on this, Carl Sigmund has written several ones, and in particular there's one that is called The Calculus of Selfishness of 2009. So this equation is actually equivalent to a Lotka-Volterra equation, and in some cases it can be solved basically if you can compute explicitly this term of what is the payoff difference on average at the population level. So which kind of outcomes you typically have for this equation is, okay, you can probably all solve it without any problem if I give you this. It's an equation where x is bounded between 0 and 1. And the typical thing that happens, if you have that groups are formed randomly, then basically your average at the population level is the same as within each one of the groups. Therefore, you will have that PC minus PD is negative, which means that your evolutionary dynamics, independent of which fraction of cooperators you start from, this will lead you to this fixed point that always exists, and that this will be stable. You have another fixed point that always exists here in 1, and this will be unstable. So basically, randomly formed groups, there's no hope for cooperators. The frequency of cooperators over evolutionary times will go to 0. Now what happens in most models that propose solutions to this evolution of cooperation is that they get a payoff function that is something like this. So there is an interior fixed point, and this often is unstable, and this one becomes stable. So basically here you have a positive payoff difference. This means that in this equation your frequency will increase because these two are always bounded between 0 and 1. And so you will have a threshold, basically, a threshold of frequency of the cooperators that is necessary for cooperation to invade. So if this threshold is relatively small, finite size fluctuations can lead you above the threshold, and once you're above the threshold, you will go to this other equilibrium. And once you're here, fluctuations will not be important, and you will just stick there. This is why most solutions that have been proposed for the evolution of cooperation work, but they work only if this threshold is small enough. Now, I see that I'm going much more slowly than I expected, so I'll just come back a moment to this picture. And I want to stress that in all these models, in most of these models, you assume that this macroscopic level of description already exists. Whereas if you want to explain the evolution of groups that are cooperative, for instance, multicellular organisms, you need also to explain how this organization first came about. So in this case, you will need somehow this groups to emerge to some extent, maybe independently of cooperation or maybe connected to cooperation. And this is something that people have started recently to deal with. making different assumptions on the way groups are formed and how does group formation couples or not with the properties of cooperating. So for instance, in a series of papers starting from 1999, Laetitia Aviles and co-worker have studied populations of units, each of which carries two traits. One trait determines whether they will be or not in a group or their propensity to join a group, and the second trait is a cooperative trait. Now in this kind of models, it turns out that you can evolve cooperative groups. However, being in a group is not costly. It's just, I mean, it happens to come for free. And this is perfectly fine, but in just intuition says that some of this grouping might bear some cost for the individual, because basically you are restricting yourself to a smaller space. There will be more competition for nutrients. Think, for instance, to the evolution of multicellularity. It might not be so good to be close to other cells that are that are similar to you. Therefore, what we have been doing is to consider cases in which a group in itself is costly. Now again, you need to do some assumptions, and what we have been considering is that you have a physical property, a trait that tells you, that plays a role both in group formation and in the role you play in a public good game. So let me just tell you briefly the kind of models we have been considering. So these models are meant to describe how groups evolved. So let's say you start from a population of particles that are dispersed, and then these particles, sometimes they can form groups, and these groups can actually be of different size. And if you have two particle types, they can also be of different composition. So let's say you have, again, these two types, then this will, well, I haven't written the same number of particles, but the important thing is that you start from a global pool of particles. By some mechanism, you group them into groups of assigned size, and then once you have done this, within each of the groups, you can play the public good game. And what our hypothesis is that basically your propensity to form aggregates is related to the propensity to cooperate within a group. So let me give you an example. If a cell has to produce a glue to stick to other cells, then the production of this glue is costly in energetic terms. Once the glue is produced, the cell will stick to others with higher probability. This also in the absence of recognition, just because it's stickier. And then the groups that will be formed will naturally contain probably cells that are not sticky. Of course, depending on the composition of the group, you will have that the whole clump of cells will be more or less coherent. So if you only have sticky cells, this clump will probably remain coherent for a long time. If you have a clump of loose cells, this will probably not be of great use for its collective function. So what we have is a cycle between these two phases. This cycle resembles very much the life cycle of species like Dictyostelium, so slime molds or myxobacteria that have exactly this kind of life cycle. So they live in isolation, then they stop dividing when the nutrients are exhausted. They form clumps. These clumps reproduce, and the reproduction of different strains depends on the composition of the groups. And then the spores are again redispersed in a common pool. So it's analogous to what is known as trait group models that have been used in the evolution of cooperation, introduced by David Sloan Wilson in 1974. So imagine you have this process. What matters, according to what I explained before, is first of all, what is the composition of the groups? So basically, for each group, now the groups are of variable size. So let's say that little n is the size of this group. Then what matters is both the composition of the groups and the distribution of the group sizes in the population. So let's say that after you have this group formation process, you're depending on whether you are white or purple, that I will call now social or asocial. So you will have two different distributions of group sizes. And so you can derive a similar condition for the evolution of social behavior that is again composed by a part that is due to self and the part that is due to the others. So this is the payoff of socials, average over the whole population, and this is the payoff of asocials. So you will have again a term B, but this time you have to sum over all the n larger than 1 of so d n over n minus c. And this term is due to yourself, basically. What's your contribution when you enter a group of size n, weighted on the distribution of groups that, social sees. And then you have another term. Well, this term, you can easily see that when n becomes large, if you only have big groups, this is useless. This just means that if you just have to decide on the basis of what is your own contribution to the group you join, you can do something if the group is small, but if the group is large, your contribution just vanishes. And then you have another term that is, again, the sum for n larger than 1 of ds es minus dA EA, and this term just tells you that you combine the distribution of group size with the composition of the groups in a way that even if the composition of the groups is exactly identical for the two cell types or player type, then you can still play with the distribution of groups in order for to change, to reverse the evolutionary dynamics with respect to well-mixed populations. So I now go briefly to an example. This can be actually calculated in some cases analytically, but for very simple models. What we have been doing is try to simulate a group formation process that resembles more or less this one. This is Dictyostelium discoideum. Cells are aggregating, and then they form mounds that turn into slugs. And these slugs are those that form eventually the fruiting body. So the typical setting in which this is observed is that the cells are spread uniformly on a surface. And how did we model this? We consider that each cell is a self-propelled particle. So it moves at constant speed. It has basically a directed random motion, which has been shown for dictyostelium cells. And then cells interact locally through some forces, an elastic force. So this force is, well, these are just the equations. This is the system, a system that has been studied by Hugues-Chatet. The only thing is that here we don't have the alignment term. We just have the force, the elastic force between the particles. Basically, according to this elastic force that I'll soon show you what is its form, cells can stick to each other. And then there is, of course, a noise component in this. So the elastic force is simply defined by that there's a hardcore potential so that particles cannot cross. And then you have, let's say, a maximal radius in which they can sense another particle. Now, if you simulate this with two particle types that have two interaction forces, after a while you have that clumps of particles are created. And what is interesting is that there are many that remain outside and they keep moving. This, of course, depending on the parameters. So once you decide that your aggregation timescale is over, you can identify the clusters, and within each one of these clusters, you can do exactly what I was saying before. So you compute how many clusters are there of a given size and what is the composition of each cluster. And on the basis of this, you can compute what is the average payoff of socials or asocials that are playing a public good game if they belong to a cluster. If they don't belong to a cluster, they just do nothing if they are asocials, or if they are socials, they pay a cost. Yes?
Why are there two distribution functions for NDS and DA?
Because they don't need to be the same. They, in a well-mixed population, they would be the same. But
aren't they just the distribution of the cluster size?
No. This is the cluster size that a social meets. And the cluster size of the groups that you just single out all the groups that contain socials, and this is the distribution of the cluster size. And this is, you do the same for the distribution of the clusters that contain asocials.
But when each cluster contains mixed asocial and social particles as in this figure,
yes.
how do you compute those two distributions?
It will be in both distributions. I mean, it will make part of both distributions. I make an example. If you have a process of group formation, again, that allows socials to stick only to socials, then this one will be a distribution of group sizes, and this one will just have a singular component in one, because asocials will not belong to any group.
Information of how the two types of particles are mixed together in a given cluster. Isn't the information redundant? I'm sorry. This is just if you take out all the clusters that are of size n. So what is the fraction of basically cooperative or of socials that you find there? So it's basically you have a distribution both in group sizes and in composition. So it's like you have two dimensions. So, but this is, I mean, is somehow a formula that you need to specify actually your group formation process to know this. So it's somehow a triviality, this one, because if you don't say how you form groups, you don't know neither D nor E. So let's say in this case, since we have built the model, we know how groups are formed, we can compute both D and D. But here it's just a numerical simulation. Basically, the algorithm, you stop the...
But in this example, you see that these clusters are actually heterogeneous. And they are
Yes.
So
yeah.
somehow the sort of mean or average quantities might not be the proper, might not capture the proper dynamics if you are lower.
Well, we average, you know, for... Yeah, so the question is, how is it that you... So since groups are composed by different particle types, how do you know that the mean is representative? But the mean is actually computed for each particle of the two strategies. So you take each particle that is social, and you average its payoff within each one of these clusters. So this is basically the original idea for evolutionary biology. You have individuals that are reproducing. So what matters is the payoff of each individual in the social context that has emerged due to the interaction between the particles. So basically, it's quite intuitive if you think of it. If you think that the function of this cluster is to let these two particle types reproduce, you just average what is the payoffs of red and blue particles within all the clusters. And then, of course, you have to take into account in the evolutionary dynamics also the fact that some particles remain outside and that they are not actually part of the collective function. I mean, they're just not part of collectives. So basically, you can update the frequencies according to your average payoff of each particle, of each particle type, sorry. And in this way, you start again another simulation by dispersing all the particles. And what you have is that for some parameter values, you start in a situation in which your clusters almost not form. You have basically a sort of weakly clustered particles. And then through evolutionary time, you end up in a situation in which you have well-defined clusters that are composed mostly of more sticky particles. If you look at it on the ecological time scale, that is the time scale on which groups are formed, you have that down here, basically you have a sort of a gas phase, and some little groups are nucleated by the red particles. Whereas here, most of the particles are inside clusters, but they are still moving around, some of them between clusters. So these are the two distributions of group size for the two strategies. So you see that asocials are found much more often outside clusters than are social particles. And the fact that red invade depends on two statistics on the population. One is again assortment, so what is the probability of meeting other socials within a group? But then what matters is also what we have called volatility, that is just what is the importance of the gas phase in this kind of picture. So how many particles remain outside groups depending on what is their properties, their social traits. And then based basically on these two quantities, you can interpret this diagrams in which we have changed the parameters of the system. And you can see that actually there is some cases in which groups of social individuals can evolve, but then there are also cases in which just no group evolves, and cases in which you have groups of asocial individuals that evolve. What is interesting is that if you want to obtain the larger possible cluster size, you need to be social. And again, this is for changing density. You still have some intermediate values of density by which you can evolve at the same time sociality, that is, let's say, the cooperative behavior in the terms of what we said before, and the structure of the population that is grouped, so that has some macroscopic entities you can see and measure. So how does this relate to microbes? There is some qualitative agreement just in the argument. For instance, what is known is that if you alter spatial structure, you affect not only the function of the collective, but also the way the groups are formed. For instance, in myxobacteria, if you let cheaters or defectors invade, these cheaters are, there are some variants that are not able to swarm. So the way their group is formed is actually tightly coupled with what is considered to be their social function. Another thing is that for the example of differential attachment, as is well known for differential attachment, how it has been studied in, for instance, for tissue reorganization, you can have, even though this is actually a snapshot of a transient, is not an asymptotic state, you still can have the emergence of a spatial structure whereby the less sticky particles are outside, and this is something that corresponds to what people have seen in experiments, that those that are considered to be asocials are sort of separated in the physical structure from the social ones. And the last point is that very recently, so basically this year, this paper is now, has just been published, and there's some ongoing work in Princeton about this. People started realizing that looking at the macroscopic aggregates is not enough. You also need to say something about the cells that remain outside the groups. This is, for instance, in the case of Dictyostelium. It turns out that a considerable amount of cells, depending on the conditions, may not join the groups. So again, if you average over the whole population, you have to take these ones into account as well. So I want to finish briefly, just going back to the original, so to the things I discussed at the beginning. Basically, what is the evolution of a collective? In this case, I have shown you, you just have some individual level properties. These properties are selected or not, depending on the emergent population structure that they trigger. So if I'm more sticky and therefore I tend to be more often into groups, then I can get some advantage of being in a group. You don't need at all to suppose that there is any collective level evolution. So just particle level evolution is far enough to let collective properties evolve. However, if you look at, for instance, the evolution of group size in this system, this is so different generations, and this is the distribution of group size. Basically, each cut of this is the picture I showed you before. So you have groups of different size. If you knew nothing about the process that leads from one generation to the next, you may conclude that this is a process in which a larger group size is selected. And actually, if you apply some of the statistical methods that are commonly used in evolutionary biology, this is the conclusion you would get. You would get to the conclusion that there is some evolution acting, some selection is acting at the collective level. However, groups that are in this component of the distribution, they have by no way generated the groups that are in the same component at successive generations, because groups are destroyed at every generation and reformed. So there is no collective level heredity. So the collective does not obey the conditions I discussed at the beginning. A collective cannot generate another collective. It can only reform, so collective reform at every time. If you think in terms of genetic relatedness, a cell that belongs to a group of size n has not a higher probability than any other cell to be in a collective of that size the following generation. So this means that you have a system in which you would like to think that the collective function has evolved because of its collective function, but you need to go through the single cell level to explain it, which is what many people like in evolutionary biology. They say eventually you can just boil everything down to the properties of the lower level replicators. Now, there are some other points of view, however, and people started realizing that if you don't have a process that is as simple as the one I have described, but on top of particles reproducing inside groups, groups can reproduce themselves, then this can change again the picture. And actually, recently, Debely and co-workers have proposed a general theoretical framework that takes into account both reproduction processes occurring at the particle and at the collective level. That are, again, it's a very general formulation, which means that for applying to specific cases, you sort of need to know how collectives beget collectives. And this has been discussed also by philosophers of biology like Peter Godfrey Smith. If you're interested, this is a very nice book in which he describes how different modes of collective reproduction are compatible with the process of natural selection. Because we are used to think to macroscopic organisms that reproduced by a single cell bottleneck. But this is not always the case in nature. You have many weird organisms that make strange things. And if you want to have a point of view that is more related to evolving these collective properties in a lab experiment with microbes, I suggest you read this paper that has just come out recently. And there will be a talk tomorrow at the Department of Biology by Paul Rainey at 11 who will speak about this. If any of you remains around, you might be interested to follow it. So, and this is more from really the point of view of experimental evolution, looking at what microbes do in a context in which they have both the possibility of reproducing as individuals and as groups. And okay, that's all. I'll just thank my co-workers. So Thomas Garcia, he made his PhD with me. He finished last year, and he's basically behind most of what I have presented you today. On the self-propelled particles, we have collaborated with Leonardo Gregory Brunet. Paul Rainey, I discussed with him on the more, let's say, philosophical issues. And Guillaume, he was a student at École Normale. I didn't present you today his work, but if you're interested about the evolution of a continuous trait that calls for stickiness, just ask me. And thank you very much for your attention. Yeah.
Yeah. Group selection, you just said that your offspring happens to be close
Yeah. Yeah, no, that's a way to create this assortment. So he was probably talking about at the collective level, so at the individual level, if you have, for instance, that your units are arranged on a lattice, and then let's say you have a lattice and you have cooperators and defectors again, and your cooperators will reproduce in one of the next sites available. Therefore, you might have clusters of cooperators that interact with themselves more than with defectors. So this cluster of cooperator will get some benefits. It will be invaded by defectors on one side, but it will manage to persist. So you have some special patterns changing in time, like spirals, I mean, all this typical pattern formation things. You get coexistence of defectors and cooperators in this case. And in the case of Baran, I don't know whether he talked about this, but when you have two levels of organization, what can happen in a more, probably more realistic cases is that these groups, they persist for some time. So therefore, you have also a dynamics within the group. So what will be your outcome in terms of fitness is not just given by the composition when the group is formed, but it will be amplified somehow by the process of reproduction within each group. This has been also, Erwin Frey has considered this kind of models. In some sense, it helps cooperation because you readily see that if you have a group that is composed mostly by defector, this will die. Whereas if it happens that you have a group that is composed by only cooperators, this will thrive forever because you don't have injection of new defectors. So basically this biases more things towards cooperation.
In your case, why do you need to destroy the group under the problem?
Well, it's just because it's the worst case scenario and it's some framework that has been used and that corresponds to dictyostelium, that is the main biological system we want to apply it to. So you don't strictly need to destroy them every time. Yes?
You said that you have some kind of alternative to Lewontin hypothesis concerning the evolution that wouldn't induce, that wouldn't speak about fitness?
Yes, yes. I can give you the reference. Actually, I think it was there before. But what we propose is that instead of so other three criteria that should be necessary conditions for evolution by natural selection. So the first one is identity. Basically, you need a criterion to identify what is your macroscopic level of description. It is somehow already hidden in Lewontin conditions. The second one is recurrence. So you need your collective level to recur at different times. And this is something more general than just reproduction. You just need to be able to identify your collectives for several successive times. And the third is genealogy. So you need to be able to determine at the collective level some kind of a genealogical relationship in order to define what you mean by heredity. You can do it if you have two levels of description, and let's say at the lower level, you know how to, what's the genealogical relationship of particles at the lower level, you can define genealogy at the higher level, presumably by knowing, by averaging the genealogy of the composing particles.
But here, the asocial particles also have identity recurrence and genealogy, and in the end they don't evolve, they just disappear.
Well, no, they all, so both particle types here evolve by natural selection. I mean, evolve by natural selection can also mean that they get extinct. That's perfectly fine with natural selection. It's just that you have a process that selects one population. Yes?
Usually, the rule of Hamilton is used to explain the cooperation between ants
Yeah.
and a lot of other cooperations. And it's used to be a necessary condition most of the time. And do you know systems of biological individuals that that apply cooperation, that form groups of cooperators without without the huge benefit of the fitness given by the gene selection?
Yeah, well, this is an example because, Dictyostelium and Myxobacteria are examples in which, in principle, you can form groups with everybody. I mean, they don't need to be kin. Of course, if you are kin, that they are present locally, they will aggregate between kin. So this, in this process, since every time you reshuffle all the population, you don't have relatedness between particles that belong to the same group. And the same can, in principle, occur for Dictyostelium. And indeed, in natural settings, Dictyostelium fruiting bodies, they are known to be composed often by more than one genotype and up to 10, I think, different genotypes. But in the lab, you can easily form chimeras.
But there is still a small relatedness between cells.
Yeah, of course, there is still a small relatedness. The point is, what does Hamilton's rule tell you? Because first of all, to compute the average relatedness and what is its role with respect to other factors is not so obvious in most cases, even though theoretically you can define it. But then if you have a really biological system, the question is eventually, does it really matter so much? It tells you, okay, you have a threshold above which cooperation can evolve, but it doesn't tell you how to compute this threshold, except in cases like, I don't know, for ants, in which you know the process of reproduction and you know the details of the mechanisms. And of course, not all haploidiploid ants are cooperating.
Does the mode of reproduction, like the sexual or asexual reproduction, affect complete, I mean, in what
Well,
way? Quantitatively?
it will. The problem is that as long as you think about two strategies competing, the qualitative cases that are possible are not so many. So basically, it will surely affect everything because you impose over this kind of processes. You have the process of reproduction of the group. Of course, in fully multicellular organisms, so classical multicellular organisms, where you have reproduction by a single-cell bottleneck, then what happens is that these groups are highly coherent because all one group is formed basically as a clone. Therefore, it is extremely efficient basically in separating good and bad guys, for instance. And this is one of the reasons why people think that reproduction by a single-cell bottleneck is so widespread and is so important. What we think is that it's not only that, it's also that it increases dramatically basically the genealogy, the genealogical relationship between your recurrences. If you have things that can take particles all the time and take them out, it's very hard to imagine that you can evolve complex developmental features. Whereas if you have something that remains stable throughout the lifetime of the collective in terms of its composition, then you can selection acts much more potently at the collective level than it does if you have things of different nature that are allowed in at any time. Yes?
Here, like how the populations goes, it can change the fitness landscape.
So,
Yes. Yeah, yeah.
is it included here? I mean, I didn't.
Well, we don't really have, let's say the fitness landscape is this one. Yes, what happens is that during evolutionary times, you change everything inside here. So you change the distribution of group sizes, and the group size distribution increases in time. In the examples I showed you, basically you start with small groups, and then you end up with large groups. And this dynamics of the group size, for instance, is coupled to the dynamics of the frequency. When you have a higher proportion of sticky cells, then you will have, on average, larger groups. But what is interesting is that basically you have a property that is independent of the recognition of the others, and that is as simple as stickiness that can drive towards this kind of evolutionary equilibrium.
All right, so thanks, Silvia.
Thank you.